I do have a more general interest in microfungi of other sorts, but my main focus is on Hyphomycetes or moulds, a group of fungi which I believe is sadly neglected by most field mycologists, and maybe, just maybe, I'll manage to change some of your minds this evening. So I'm basically going to talk about three things. Firstly, just a very brief introduction to Hyphomycetes and what they are. Main bit of the talk will be describing my approach to collecting and identifying them. And then in the final section, I will talk a bit about how you interpret distribution data of Hyphomycetes. So what are they? Well, as many of you I'm sure will be aware, they're basically just anamorphic asexual forms of fungi. Generally asexual forms of Ascomycetes, but in some cases of Basidiomycetes. In some instances, there's a very well-established connection between the asexual form and the sexual form of the fungus, and you may even find them growing together in the field. But there are also a very large number of Hyphomycetes in which no sexual form is known at all, or at least no connection has yet been made. And the final point I ought to make here is that this is not a taxonomically meaningful term. It's just an umbrella sort of catch-all category. It is of no taxonomic relevance in any way. So if you talk about moulds to the average man or woman in the street, it's not a term that's going to generate feelings of goodwill. It's a sort of response when you mention mould, and with good reason. You know, moulds are responsible for the rotting of many foodstuffs, such as the mould you commonly get on rotting apples. They also cause a very wide range of diseases. So this is a image of fusarium rot of cereals, which causes major economic losses of things such as wheat and barley and maize. So it's a huge economic problem for farmers. And they also even cause diseases in humans. So athlete's foot is caused by a couple of different species of Hyphomycete. But of course, rot and decay are important parts of biological cycles. It's an important role in nature. And some Hyphomycetes also have more specific relevance to humans. So penicillin, of course, discovered by Alexander Fleming in the 1920s, that is basically derived from a Hyphomycete, a couple of different species of penicillium. Different species of penicillium is responsible for the blue mould in cheeses, such as Stilton. Also, soy sauce is fermented soybeans infected with an Aspergillus mould. And then there's the noble rot, as it's called, of grapes, which is an essential component of some sweet white wines, such as Sauterne. That is caused by very common Hyphomycete, Botrytis cinerea, which leads to the sort of raisinifying, if that's a word, of grapes, makes the wine much sweeter and adds flavour to the wine. So they are of interest to humanity in various ways, but certainly they are not just a problem for humanity. So in terms of structure, in very simple terms, this shows the main components of a Hyphomycete. You've basically got a more or less specialised hypha called a conidiophore, and that bears the reproductive structures. So the asexual spores or conidia, which arrive from conidiogenous cells. So in very simple terms, those are the basic components of a Hyphomycete. And we're talking small here. That whole structure there on that slide is about 50 microns in length. So they're not big. So a very simple example, I'm just going to whiz through a few more examples of Hyphomycetes under the microscope to give an idea of the diversity of forms that you get. So you can get these very richly branched structures in Hyphomycetes. So there's a species of Brachysporium on the left and Goniptricum on the right. So these branched moulds often form sort of wefts or mats on rotting logs. You may get species in which the conidia aggregated together into fairly dense heads, which is quite distinctive. So, for example, the image on the right there is of a periconia species, which is very commonly found on rotting plants. The examples I've shown so far are all what are known as dematiaceous Hyphomycetes, so they've got a brown pigment, but there are a lot of Hyphomycetes without pigment, so hyaline species such as these. You also get some species that have various, say, shades of yellow or orange or pink, at least when you've got an aggregation of spores. Then you also get forms in which the individual hyphae or conidiophores are aggregated together into what are called cinemata. So that makes a more solid structure. Also, an interesting facet of some species is that you get very prominent non-fertile structures, or setae. So the example on the left here is a Gonyptricum species, in which you've got these relatively long setae. They're probably about 500 microns long, and they are the most prominent part of the fungus. They're the part that you can just about make out with the naked eye. The fertile structures are absolutely minute, and you can see that in the inset photograph in the middle there, where you've just got this tiny flask-shaped conidiogenous cell with a conidium developing at the base, at the apex. And these are found just at the very extreme base of the setae. Then on the right, you've got another example of a seta in a Hyphomycete with a swollen vesicle at the apex. So this is a species of Cylindra caladi. A very important feature of Hyphomycetes, very important in terms of identification, is how the conidia are actually formed. So perhaps in the most simple type, the conidium is just formed at the apex of a conidiogenous cell and basically just breaks off. So this is an example here of a Sporidesmium species, and when that conidium there is fully mature, it will just break off from the conidiogenous cell. But you sometimes get additional slightly more complex structures. So you might get these raised scars or swellings in some species where the conidia develop. So this is a species of Pseudospiropes. Or you may get these cylindrical denticles on which the conidia were born. Or you may get other rather specialised forms of conidiogenous cells. So these are two different examples here of what are known as phylids. So these are specialised cells with an open apex, and they produce a variety of conidia in sequence, which are then discharged through the apex of the phylid. So the example on the left there is a species of Torula, a big genus with a wide range of species that you'll often find in the field in the UK and elsewhere. Then you may also get species where the conidia are extruded or blown out through pores in the cell wall. This is something that I really do struggle to capture adequately in photographs, but I think you can just about make it out in that example on the left, where you've got a conidium that's formed on the left-hand side of the cell, and you can just make out the pore that it's been extruded through at the base of the conidium. And then on the right, you've got an example of another species where you can make out those pores at the apex of the cells, and the conidia are, as I say, blown out through those pores. But for me, one of the great delights of Hyphomycetes is the sheer diversity of conidia that you get. They are much more spectacular, in my humble opinion, than those of Basidiomycetes. I mean, yes, you do get some that are really pretty boring, but you also get a range of different forms, such as those shown here. So very heterogeneous in terms of their shape, their size, their colour, and so on. And often very, very distinctive. In some cases, you genuinely only need to see one spore and you can identify at least a genus and sometimes the species. So there's a range of different conidia there, and then you also get even more complex structures. So you can get branched conidia, conidia that form helices, such as the top right example there, and so on. I mean, these are, I think, amazing structures, and it's interesting to speculate on why they may have evolved to form these kinds of shapes and forms. So that's a quick run through what Hyphomycetes are, and some images to give you an idea of the diversity of forms that you encounter. So I'm now going to talk about the approach that I take to actually collecting and identifying these things. It's broken down into 3 phases. So first of all, how do you go about collecting what are very tiny, genuinely microscopic fungi? And the main point, of course, is that you're basically not even going to notice these things if you're sort of casually walking through a wood, because they're so small. Even if you do find them, you're almost certainly not going to be able to identify them. So that does pose particular challenges. So the kind of approach I take is to, as far as you can, try to sample the diversity of the site you're in. And you can define that diversity in a variety of different ways. So it could be different habitats within a site, different microhabitats, a range of different plant species, and even within one species, a range of different substrates. And each of these will normally support a different assemblage of Hyphomycetes and other fungi. So I'm not talking here about taking a fully systematic approach, but basically just trying to collect from a range of different situations within whatever locality you're at. The main thing I'd want to emphasise is the need for patience and persistence. And I love this quote from Dennis in British Ascomycetes. So he wasn't actually talking here about Hyphomycetes, but it's just as relevant. So he's saying that, one of our most successful collectors of Halotiales, which are Discomycetes, makes a practice of spreading a Macintosh in an attractive looking ditch, lying upon it and not stirring further for an hour or more, while he methodically scrutinises every piece of vegetable debris within reach. I mean, for me, this first of all conjures up the question, what on earth is an attractive looking ditch? I mean, attractiveness is not a feature what one normally associates with ditches in any way at all. But it is actually a good way of collecting these minute fungi. I must say I very rarely spend as much as an hour in one place, but it's quite common for me to just sit in one spot for maybe 20 minutes or so and just look at the range of leaves and sticks and so on in the vicinity and scrutinise them to see what Hyphomycetes might be present. And as an example of how persistence and repeated recording can pay off, this is a fungal hotspot in Penrith where I live. It's my back garden, which is a small, pretty unremarkable plot of land. I mean, the only thing it's really got going for it in terms of fungi is that it's pretty unkempt because I don't look after it very well. But it's certainly not somewhere that most people would spend much time looking for fungi. But of course, because I live there and I can go out every day and different seasons and so on, you gradually accumulate quite a list. And in the 18 months or so I've been living here, I've now recorded 40, I think is the latest total, 40 species of microfungi in this one pretty small area. Most of these, I must say, aren't Hyphomycetes. There are a range of mildews, rust fungi, a few Hyphomycetes on rotting vegetation and so on. But it just shows that even in the most unpromising habitats, you can find an awful lot of different species. And 3 of the species I found in the back garden have very few records in the UK, simply probably because people aren't looking for them. Now, most of the Hyphomycetes that I collect are associated with often decaying vegetable matter. In some cases, they're parasites on living plants. So ideally, you really need to be at least an adequate botanist. So having access to good reference materials such as Stacey's flora is important. But of course, you don't always have the luxury of actually having to identify plants that are in good condition and flowering. You're trying to identify dying plants often. So a book such as this, The Vegetative Key to the British Flora, is absolutely invaluable, a wonderful work, and really does help to identify plants that don't actually have flowers. It's also well worthwhile trying to get the knack of being able to identify wood as well. Sometimes you can make some assumption about what kind of wood a branch or a stick is, but it's often pays to check. And there's a pretty good and pretty simple key to some of the common species that was published in Field Mycology a few years ago. So that's some of the challenges associated with collecting in the field. Just as a slightly light-hearted interlude, one of the other challenges that I have faced, and some of you may do as well, is going out in the field and collecting fungi with your dog. When I got my dog just over a year ago, I fondly imagined it would be sort of welcome companion on my field trips, but it hasn't proved to be quite as easy as that. Quite often you'll sit yourself down in a spot and the dog will decide that perhaps that wonderful specimen you want to look at, he's just going to pee on. That's what they do. Or you'll be picking up a stick, wanting to look at the stick to see what's on it, and the dog will decide that that stick is absolutely the stick he has to have this moment. So you give him that stick. You pick up another stick, and then of course that's the stick he really wants above all other sticks. So before you know it, you've handed over a whole range of promising looking sticks to your dog and haven't looked at any at all yourself. But perhaps the biggest challenge is that basically for a dog, it's a pretty boring activity. You're sitting there with your human while they look at fungi. So they can be tempted to just go off and do other things. And in the case of my dog, that normally involves chasing rabbits or squirrels or deer. And on the recent BMS foray in Bedfordshire this spring, he did head off chasing some muntjac in the wood in mid-Bedfordshire and wasn't seen for another hour and a half. So beware taking your dog out with you looking for fungi in the field. Though, as I'm sure Carol would say, they do have at least the potential to be trained to sniff out truffles and other hypogeous species, and that's not a knack I've yet managed to impart to my dog. So, assuming you've collected your species, they haven't been eaten by your dog, you've got them back into your lab, how do you go about actually identifying? Well, the good news is that you really don't need many specialist techniques. You don't need to be cutting sections of things. You don't need lots of different chemicals and stains. All you need to do is get a bit of your mould, your Hyphomycetes, slap it on a slide in a bit of water, put a cover slip on top, and there you are. That's basically all you need to do. The challenges are mainly around the minute size of the material you're dealing with. And you've really got to be competent and comfortable with handling such minute material. You've certainly got to be able to use oil immersion to get up to 1,000 times magnification, or you won't be able to see the features, the diagnostic features that you need to see. And being able to interpret some of those features does take quite a lot of experience, I found. In particular, you need to be looking at the form of the conidia and how the conidia are developed. So the different types of conidia genus cells that I mentioned earlier. And of course, in all the drawings and all the books, these things look clear cut. When you're actually looking at them in real life down the microscope, it can be very difficult to actually interpret what you're seeing. And that, as I say, does take quite a bit of experience to get used to. Now, the approach I take, at least with material that I don't know when don't know the name of when I've collected it, is really making pretty comprehensive records of the material before I try and identify it. So you've collected a species. This is Spondylocladiopsis cupulicula, which grows on beech cupules. So what I do with something like this is actually, before I try to identify it, I do a fairly full description. So I look at the conidiophores, I look at the conidiogenous cells, I look at the conidia and describe what I'm seeing. You know, what size are they, what shape are they, what colour are they, and try and do that without any prior assumptions, so that I'm looking at each specimen as objectively as I can. And then once I've done that, I'll try and key the species out or identify it using various reference material. And I find this approach, although I must admit it is time consuming, it does work well in really forcing you to look closely at your material and describe what you're seeing. I don't actually take this approach now with everything that I collect, but I do still do this with a lot of my material. And apart from helping to give you that objective record, you do find once you've been doing it for a few years, you'll build up a very useful sort of database of material, of descriptions of the same species. So you can see how, for example, the conidia vary between different samples. You will, of course, need access to a range of reference material. I mean, two of the standard works, which I'm sure some of you will be familiar with, are Ellis's Dematiaceous Hyphomycetes and the imaginatively titled More Dematiaceous Hyphomycetes. These are really good books and well worth investing in. And a lot of the material is summarised in the Microfungi on Land Plants book. If you can afford it, I would also highly recommend this book, this really magisterial volume, The Genera of Hyphomycetes. Has the most wonderful illustrations in it. And I must confess, on many occasions, I have been reduced just flicking through the pages of illustrations, desperately trying to find something that matches the specimen I'm looking at down the microscope. So a range of books, all kinds of scientific papers, of course, which you'll be familiar with from looking at different types of fungi. And also increasingly, it's worth looking at internet resources. So there are various fungi forums such as ASCO France, which is very good. And also, of course, our very own BMS Facebook page. There are real experts there who are often very willing to give up their time and to give you some advice on problematic specimens. One other point I want to emphasise is that you really shouldn't believe everything you read in the textbooks and the scientific literature. Things change. People make errors. And of course, I mean, I'm not talking about the huge changes in taxonomy and so on, which are always occurring, but basic errors in description. So that, again, is a good reason why it's worth looking at things with a fresh eye and no preconceptions. So I'm just going to give a couple of examples on how you could be led astray by looking at Ellis's volumes, for example. So one of the strengths, but also one of the weaknesses of Ellis and Ellis microfungi on land plants is that basically you get into the fungi species by identifying the host plant. And that's great in some circumstances, but it can lead you down the wrong path. So this Hyphomycete, Gyrothrix podosperma, which has these lovely dark brown CT with curved apices, is described in Ellis as being found on dead carex leaves, dead sedge leaves. That, in my experience, is not its most frequent habitat in the UK at all. I think it's much more frequent on dead tree leaves, especially trees that have fairly tough leaves, such as holly or holm oak and so on. That, I think, is its typical habitat, but obviously, for whatever reason, the Ellises didn't find this species in that habitat. They just found it on carex leaves, so that is where you'll find it in their book. But you would have an awful job if you find it on a holly leaf, identifying it just by reference to Ellis's volume. So the ecological information in published material may not be accurate. In some cases, the descriptions may also not be entirely accurate. So Ellis also describes this species, a very common species of decaying wood, Cylindrotrichum oligospermum, and he says it doesn't have CT. Well, actually it does. It has these very prominent long CT which form a tall layer above the shorter conidiophores, which bear the spores. And that's quite a distinctive feature of this species. But again, you won't get that from looking at Ellis's books and papers. So, as I say, it pays to look with a fresh eye and just have a degree of skepticism about what you read. I should say in no way is this a real criticism of Ellis's volumes. They are remarkable works, and it's just absolutely inevitable in any published descriptions that there will be some errors and oversights. Okay, so moving on, hopefully you've managed to identify or have a stab at identifying your fungus. What are the next steps? Well, for me, an important step is actually keeping a collection of specimens. Dead easy to do, they're very easy to dry. You can just air dry these specimens, pop them in an envelope with the details on the front, and then store them in cardboard boxes. And I find this very useful in that you can then refer back to this dried material and compare it with other material you collect at a later date. It also, of course, gives you specimens that you can pass on to national and local herbaria if need be. One of the things that really surprised me when I first started looking at microfungi in general and Hyphomycetes in particular is just how many specimens you're not able to put a name to. So I come from a botanical background looking at flowering plants and bryophytes, and you can name 99.9% of the material you collect with absolute confidence with those groups. Certainly isn't the same with microfungi. So you gradually accumulate a pretty large number of specimens that are either unnamed or have very provisional names. And the only way forward really with these is, I think, to start culturing them to get decent material. And then, of course, to start doing some DNA analysis, which, of course, the BMS has been encouraging field mycologists to do in recent years. And that really is, I think, the way to try and unravel some of these problematic specimens. And then finally, I would make a plea to everyone to make your information available to others so that they can look at it, learn from it, and challenge it. And the Fungal Records Database of Britain and Ireland, FRDBI, is the mechanism that I use to do that. I put all my records on that database. And I don't think it matters if you've got the identification. If it's only provisional, you can say it's a provisional identification. You can add your text descriptions, you can add photos, and then the information is out there for others to look at, which I think is absolutely vital. So that, in summary, is the approach that I take to collecting and identifying Hyphomycetes. Of course, in practice, it's nowhere near as easy as I've made out. You will end up spending many frustrating hours both out in the field and peering down a microscope. But I hope if you do have an interest in looking at these minute organisms, my tips will give you some pointers as to how to approach it. So in the last part of the talk, last 10 minutes or so, I want to talk about how you can interpret the distribution data, particularly for Hyphomycetes, but this applies equally to other microfungi as well. So the UK is probably the best studied country in the world in terms of its biodiversity. We've had very long and rich history of people recording wildlife, plants and fungi in this country. But fungi, and especially microfungi, are very badly under-recorded compared to many, many other groups. And the reasons for that are very familiar, of course, to all of us. I don't need to go on about those. So they're very under-recorded. And also, of course, the data that we have available, for example, on the FRDBI, is going to be affected by various biases. And I just want to give some examples of that in the next few minutes. So first off, let's look at a very common Hyphomycete. So this is Torula herbarum, which is a species that's often found on decaying herbaceous stems, so big plants such as hogweed or nettle, for example. You'll find this forming black velvety patches at the base of the stems. Very common species. And when I downloaded the map from FRDBI, I was actually pleasantly surprised to see that the map does show indeed that it is frequently distributed more or less throughout Britain. Though it's very interesting that there are so few records in Scotland. Is that really the case? I don't know. It may be true, or it may just reflect the fact that very few people have been recording these small microfungi up in Scotland. Another example, which really does illustrate some of the biases in the data, is this parasite, Ramularia septata, which is found on dead endymion leaves. Sorry, not endymion, snowdrop leaves. So it forms these brown tips to the leaves, and if you look very closely, you'll see a white fuzz, which is the fungus. If you download the distribution map from FRDBI, you get this very patchy distribution with clusters of records in different parts of England and Wales, mainly. So you think, well, what's causing that? Is it something to do with climate or geology? Well, no, it basically, as far as I can tell, just shows where the relatively small number of people are who have been identifying this fungus. So you've got a cluster of records down in the Gloucestershire Somerset area, which is mainly Mick Legon and a few others. Cluster of records in Mid Wales, which is Raywoods and Arthur Chater, and so on. This species is probably on snowdrops throughout certainly England and Wales and possibly Scotland. But again, only a limited number of people have been recording it. So you get a very misleading impression from the distribution data. A couple of interesting examples. First one is this species, Anavirga laxa, which was first described by Sutton in 1975 from the cupules of Castanea sativa, sweet chestnut. And he described it as being frequent on that particular substrate. There are, I think it's about 18 records in the database. Almost all of those are from the 70s and 80s. There's a couple from the 1990s, no records at all from the 21st century. I've never seen this species, and I have actually spent quite a lot of time looking at the cupules of Castanea for microfungi. So what's going on? Has it actually genuinely declined in the last 30 or 40 years? Or is it just that I've got a blind spot for this fungus, which is entirely possible? But we simply don't know. And it'd be fascinating to find out. Then you also have examples of the converse scenario. So this is a lovely little fungus called Chaetopsina fulva. It's one of the few Hyphomycetes that you can identify with some confidence in the field because it has these tall setose conidiophores that are a bright pinkish orange colour. And when it's present in some abundance, it's really quite striking. First found in Britain, again in the 1980s, a couple of records over the next few years. And then almost all the other records are in the last 5 years or so, when I've been finding it in a sort of swathe of southern England from Suffolk over to near Bristol. But I seem to be the only person who's finding this fungus. So again, it raises questions. Is this a species that is increasing, expanding its distribution, perhaps because of climate change? Well, that's possible. Is it just that I happen to have now got my eye in for it and I'm specifically looking for it so that I'm finding it more frequently? Again, we just don't know. So it's very difficult to interpret the data that we have available. And then just some examples of where there probably are some genuine geographical patterns in distribution. Dictyochaeta setosa is a species of decaying holly leaves. And I think this genuinely has a sort of southern and western distribution in Britain. It's certainly relatively frequent in parts of Devon when I've been looking. But I don't find it at all in eastern England or in northern England, although I can see there is one record there from up in Yorkshire or Cumbria area. So I think here we have a species that, although there's relatively few records, genuinely does have a distinct geographical pattern to its distribution. Another one, which I think is probably truly a northern species in Britain, is Brachysporium breve. Only 3 records, so it's difficult to infer too much from that. But I think if this species was at all frequent in southern Britain, people would have been finding it, because it doesn't have a particularly specialised habitat. So I think there are some species such as this that really do have a northern distribution. So that's some thoughts on the distribution data of these Hyphomycetes. Then my final slide really, I guess, has an element of despair to it, which is the more you look at these things, the more time you spend studying them, the more time you look at the, well, literally 2.5 million records of fungi on FRDBI, the more you realise actually how little we know about them. You know, what do we really know about where these things are, how they're responding to climate change and so on, what their ecological preferences are. So what's the point? Why do we do this? And can we actually use the data to inform decisions, for example, about conservation? And certainly there have been times when I've thought to myself, why do I do this? But, you know, I quickly reconvince myself it is something I enjoy. I mean, for me, I just get a kick out of going out in the field, collecting these things, and the challenge of identifying them. That's enough of a motivation for me. And I genuinely believe that in my own very small way, you know, you are contributing something to the body of scientific knowledge. Well, that's what I tell myself anyway. But we have to be realistic. You know, we don't know much about many fungi, certainly very little about microfungi. So can we really use the data we have available to inform decision making? And I think we've got to be realistic about this. In terms of biodiversity conservation, microfungi, Hyphomycetes, are probably not going to play much of a part at the moment because we don't know enough about them. There are guidelines now for the selection of sites of special scientific interest for fungi, but they explicitly make the point that they don't really cover microfungi because we don't know enough about them. And I think if we are to conserve the full range of biodiversity, if we are to conserve microfungi, the only way we're really going to be able to do that is by protecting the habitats that they grow in, and hopefully ensuring that by protecting a representative range of high quality habitats, woodlands, wetlands and so on. In that way, we're protecting all of the associated organisms. But that is a big assumption to make. So we don't know very much. A few final thoughts. We've got a lot of data, but we does have severe limitations. And I think it's incumbent on us to make sure that the people who actually use the data fully understand what those limitations are. And I hope I've demonstrated that, you know, you've got these biases in the data, you've got severe under-recording of many species. When we make records available, for example, to conservation agencies, it's important that they realise these limitations. But to end on a note of hope, I think there are lots of opportunities to really improve the situation by taking on board new technologies. So I've mentioned DNA analysis, which has really taken off in the last decade or so, and is becoming increasingly accessible to field mycologists. That is going to help enormously, and I think we should embrace that with open arms. Also, there are possibly new techniques for data analysis that maybe could extract some sort of pattern from the data we have. As I think I've mentioned, there are 2.3 million records of fungi we have on the FRDBI. It might be that there are some forms of analysis that could at least extract some sort of information from that, even though a lot of the records are patchy. And then I think it's vital that we do build partnerships, particularly between field mycologists such as myself and professional scientists such as those who work at Kew. both parties to that partnership can contribute a lot. And I think working together is definitely the way forward. And I think that has been demonstrated, for example, by the work that's been done recently on DNA analysis. So that is it. That was a run through an introduction to Hyphomycetes. I'm very happy now to take questions. Thank you very much.
Thank you, Marcus. I'd like to thank you for that really interesting talks, wonderful images. And if anybody has any questions, could you put them in the chat facility rather than unmute yourself so we can deal with them sequentially?
Right, I'm just opening the chat up now.
Marcus, I'll read. I can see some that we've got there already. It's Emma here. I'm just going to read through the ones that I can see. So there's a variety of questions on different topics. One about microscopy. So the question is from Angela. Why do you need to be able to use oil immersion if you put it in water and put a cover slip on?
So the oil immersion is really just for very high magnification. So yeah, you just put the material in water, cover slip on top of the water. But to get to use the oil immersion lens, the very high magnification lens, you need to put the oil on top of the cover slip because it simply won't work without it. And you can do something without oil immersion. It's not a completely lost cause with these small fungi, but it really does help if you have a microscope that does have the facility to use oil immersion.
Thank you. Flavia asks, in a time where microfungi are mostly identified based on molecular phylogeny, is the simple morphological description or identification accepted for floristic or ecological surveys?
I think that's a fascinating question. And there is no answer to it at the moment. It's a real challenge because, of course, there will be the DNA analysis will reveal species that you didn't even see in the field that may have no real visible structures that you can make out. I think at the moment the emphasis is still on the morphology and the description, and that is genuinely accepted for ecological surveys, but I think increasingly it's being combined with molecular phylogeny and DNA analysis, and that trend is only going to continue. And I don't think we should find that a problem. I think we have to accept these new technologies, but they need to be combined, I would say, with people who know about the things in the field. It's that combination of the field mycologists and the scientists who can apply these techniques that's so important.
Related to that, Eric asks, what is the coverage or quality of the published DNA sequences?
For Hyphomycetes, it is pretty scanty. There are large numbers of species that don't have data available for them. And I know people at Kew are keen to at least start remedying this, and would welcome any collections of Hyphomycetes they can do some analysis on. But yeah, they are not well studied in any way at the moment. Of course, the emphasis has often been on commercially important species such as pests and other diseases, rather than the kind of things that I'm interested in.
There's a few questions about where, which species of plants you might find the Hyphomycetes on. So first of all, a clarification, I think, is from Roger, is Ramularia in bluebells or snowdrops?
Snowdrops, apologies, that was my mistake. The species I was talking about, Ramularia septata, is on snowdrops, Galanthus species. But there are, God, I don't know how many different Ramularia species. They a wide range found on numerous different host plants. Interesting group to look at. So if you look at plants, particularly in the spring and summer, and see spots on the leaves, could be various things and not necessarily fungal, but quite often it will be a Ramularia species of some sort or another.
Okay. And a question from Sam, do you see other Hyphomycetes on chestnut cupels other than the one you mentioned? Sam says, the other week I photographed something that looks like one inside an old chestnut casing, but no microscopy, I'm afraid.
Yes, there's really quite an interesting assemblage of species that are associated with chestnut cupules, some of which are more or less restricted to that substrate. So if you look at Brian Sutton's paper, which I think I gave a reference to in the relevant slide, he gives a good description of many of the species, and they're also in Ellis and Ellis as well. So there's an assemblage of about 15 different species that you can find.
Another question, slightly different angle now. Are there any Hyphomycetes that have properties you find especially unusual, interesting, or useful, for example, medicinal or culinary uses?
Well, I mean, the ones I'm aware of, ones I mentioned at the beginning of the talk. So yeah, without Hyphomycetes, we wouldn't have soy sauce, we wouldn't have blue cheese, and I could probably live without soy sauce, but blue cheese, well, the world would be a poorer place without it. So,
similar question along that sort of medicinal lines. Are some Hyphomycetes a potential health hazard? For example, inhaling conidiaspores. Are there any reports of issues with allergies?
I believe there are some potential issues. I mean, my own approach is that the volume of material I'm dealing with, which is minute samples that you're collecting in the field, it's very, very unlikely to be a problem. But I know there are some species of mould where in household environments, if you live in a damp environment and you get mould on the walls, it can cause problems for people. So you do need to be aware of that. But I don't think it's a big risk at all for collecting them in the field and identifying them.
A couple of questions about mycotoxins from both Jane and Rinos. So Jane asks, do any of these produce mycotoxins? And then also, how do we know that the Aspergillus species used in soy manufacture, for example, does not produce mycotoxins? Also, I agree with you about the blue cheese.
Good. I'm afraid you're certainly testing the limits of my knowledge here. It's not a subject I know much about. I mean, I do know, I showed a slide of the fusarium rot on maize near the beginning of the talk. And one of the problems I believe there is not just that the mould reduces the yield of the cereals, but also that it produces mycotoxins, which means that you can't use the bit that's left, for human consumption or for livestock because it's poisonous. So that's one example. I'm sure there are many, many others, but I'm afraid it's not something I know much about.
Rajamani has asked, could you elaborate on your storage method you mentioned? If Hyphomycetes can grow on the agar medium, why should they be stored in paper bags?
The important thing is that they're thoroughly dried before you put them in the paper packets. So, as I say, just air drying them will do. You don't need to put them into a special dryer. But once they're fully dried, you can just put them in the packet and they will keep, well, for years. The big problem you will face is infection by mites. Somehow, whatever precautions you take, mites will get in somehow. So what I do is periodically put the boxes into a deep freeze, which seems to just about keep on top of the problem. But I say it's very, very easy to do.
That's mites, not mice, right?
Mice I haven't yet had a problem with, but mites are a definite problem. It's really quite distressing when you open up a packet and you suddenly see the minute bits of what you think are fungus begin to move.
One more question from Ali. How do you capture aquatic Hyphomycetes from degrading submerged stems in water?
Again, I'm afraid this isn't something I know much about. It's not an area of mycology I've ever ventured into. I think it is relatively simple to just take water samples and to then just put the water from a stream, from the foam in a stream under the microscope and see, you get these wonderful conidial shapes that you'll see. I don't know any more about it than that, I'm afraid. There are some good papers on it which might give you more information.
I've just had one more question. Someone commented, collecting drift material or driftwood would help with that. A comment in the chat.
Yeah, I mean, that's fast. Yes, I mean, that's again something that would be really interesting to look at. You'd probably get a whole different assemblage of species on driftwood, and who knows where some of them come from.
So I think that's just about it. There's a few comments saying thank you very much for the excellent reminder of pretty microbeasties. Thanks for an interesting talk. And a final comment from Flavia, who says, my degree thesis in Italy was about Ellis fungi in Mediterranean forests. I was very fond of these fungi and I studied a lot, but I had to change field because nobody, at least in Italy, was funding projects on the biology or ecology of these species, which resonates with what you were saying.
It does. I mean, if you go back to 1970s, 1980s, there were a relatively small number of people who were actively working on this group of fungi in Britain. That's simply not the case now, which is unfortunate. I mean, because I hope, if nothing else, I've shown they genuinely are. They're very attractive things under the microscope and worth looking at, if only for that.
Thank you very much, Marcus. That's been a fantastic talk.
It's a pleasure. Well, thank you all for listening.
And if anybody missed any part of it, it is being recorded and it will be put on the BMS website within the next week or so.
Yeah, and I would say if anyone wants to get in touch, if you've got specific questions or anything, I'm very happy for people to get in touch with me.
Via the BMS office.
Yeah.
Yeah, if you email us at the BMS, you'll find our details on the website and then we can pass your messages on to Marcus.
Yeah.
Fantastic. Thank you very much.
Great.
And enjoy the rest of your day, everybody.
Right.
Thank you.
Bye.
Thank you, Marcus.